morning. Welcome back here at ESCMID Global TV. My name is Judith Cohen. I am your host here at ESCMID Global TV. And I am very excited to introduce my next guest here live in our studio, who is no one less than one of this year's ESCMID Global Keynote Speakers, Mihai Netea. He's a professor of infectious diseases, immunology and global health at the Radboud University in Nijmegen, the Netherlands. And this afternoon he will be holding his keynote lecture in Hall A at 4:15 on the role of trained innate immunity in infections. So a very warm welcome, Mihai, for joining me here in the studio and taking the time on this big day for you already at the Congress.
Thank you very much.
So are you looking forward to holding your keynote lecture?
Absolutely, very exciting, yes.
Yes. So I think it's interesting for our viewers, for maybe some of them are still considering whether this is their keynote that they would like to attend or not. So let's just dive a little bit into the subject and have a little chat about that. Innate immunity in infections, it's important.
Well, it is in the end, and everybody knows that, and we feel that also as clinicians. For example, innate immunity are the mechanisms of the host defense which act in the first hours and days after an infection, when T and B cells don't have yet time to develop. So until then, we have to deal with the infections basically using our other types of cells, especially myeloid cells, granulocytes, monocytes, macrophages, and so on. And we know very well, for example, people with chemotherapy, which are neutropenic, that they will have a very high susceptibility to infections. So understanding these mechanisms of innate immune host defense against infection is crucial to understand the entire host defense.
Yeah, and the entire scala of infections.
Absolutely.
From
yes.
severe septic infections.
From severe septic infections to TB, but also to HIV. We understand more and more that also for HIV, for example, even for infections in which adaptive immunity mediated by T and B cells is crucial, also innate immune cells, such as antigen-presenting cells, have a crucial role in presenting the antigen, being able to stimulate the T and B cells. So there is no infection practically in which innate immune mechanisms are not important.
Yeah. So how many of the innate mechanisms have been unraveled so far, do you believe? Do we know almost everything there is to know, or is there still yet a lot not clear?
As always, we know much more than we knew 50 years ago, but of course there are still many things that we don't. And with every discovery that we make, we also discover also the things that we do not know. Basically, in terms of innate immune responses, we have a couple of major groups, let's say. You can call also the barrier function of the skin and the mucosa also an innate immune host defense mechanism. But even if we don't count them, we have cellular innate immune responses and also humoral innate immune responses. And the cellular innate immune responses are what monocytes are doing, macrophages, neutrophils, and so on, but also natural killer cells, NK cells, and innate lymphoid cells. And on the other hand, the humoral innate immune responses are also crucial. There are defensins which are secreted on the surface of our skin and in our mucosa. There is complement in our blood. There are all types of enzymes which are present in secretory fluids and so on. So all these are also components of the innate immune responses.
Yeah. So your field of area is really in the understanding of what lies behind beneath the infections. What do you feel are the challenges within this field of area?
So what is very important is how do we manage actually to turn this knowledge also in applicable treatments to the patients. And of course, some things we know very well. For example, if somebody is deeply neutropenic, we should do our best to reduce the period when the patient has neutropenia, and that is done with colony-stimulating factors, for example, in hematologic patients and so on. So that is already a very important component of the immunotherapy arsenal that we have during infections. But beyond that, we were not yet able to learn how to use the power of innate immune responses very much, a part of the colony-stimulating factors. So this is something that we try to understand. We also know that innate immune responses on the one hand, are is incredibly important and very crucial for host defense, but on the other hand, they can also be dysregulated during severe infections. And the most important example of that is sepsis, for example, in which innate immune responses, while very important, especially in the beginning of the process, to try to fight against the infection, it can be dysregulated, it can be overwhelmingly activated, inducing systemic hyperinflammation, which is also deleterious for the host. So there, we need to modulate these responses during sepsis. On the other hand, we also have other patients with sepsis, for example, who have immunoparalysis, where all these mechanisms do not work properly. So patient stratification, for example, for personalized immunotherapy is something that we need to do in the coming years, and this is a very important area of research for the coming period.
Yeah, and this is your line of research also, because your keynote is, of course,
well, the keynote
much
will not be about
than,
that. Yes,
no,
my keynote will not be over that. My keynote will be over the capacity of innate immune cells not to act only in the acute phase of an infection, but also to retain information of that infection and to use it when we are reinfected.
Yeah, so the trained
yes, exactly. They are trained. They use this information, which is encoded at epigenetic level in the nucleus of the cell, and they will react more appropriately the next time that we encounter an infection.
Yeah, and how large is the role or the impact of the trained innate immunity? Isn't the biggest problem or biggest key issue that the first time that the patient gets the infection?
Yes, we see basically that the activation status of innate immunity, which partially depend on training of the immune response. Another important part is genetically regulated, so we are all different from a genetic point of view. Some of us have blonde hair and some of us have brown hair, and the same is with the immune responses. Some of us have certain components which work stronger than the others. So part of the regulation of the innate immune responses is genetically regulated, but another part is epigenetically regulated, is trained during whatever we encounter during our lifetimes, and that is what we try to understand. And this makes how resistant somebody is to a certain infection. And we believe that
epigenetic, just to recall, maybe some viewers have the same question, but epigenetic is that the DNA changes, so you train the immune cells, but the DNA can be passed on to another generation, the changes in the DNA.
Well, that is the genetic. That is the genetic
and the
component.
epigenetic is...
So genetic changes are the changes which lead to a change in the order of the nucleosides in the DNA. So you have a mutation, and that you can transmit to the next generation. Epigenetic is regulation of gene transcription, which is different from change in the base pair order in the DNA. And that is regulated through either methylation of certain parts of the DNA molecule. It's also through chemical changes that take place in the histones. The DNA is packaged in our nucleus around nucleosomes, and the center of the nucleosomes is formed by proteins, which are histones. And depending on whether histones are acetylated, phosphorylated, methylated, and so on, the packaging can be different. If the packaging is very dense, the transcription factors cannot bind and they cannot transcribe genes which are important for host defense. Whereas what we have observed is that after an infection, this packaging is more relaxed. There is much more space for the transcription factors to come and bind to the promoters and increase the host defense mechanisms much better.
Yeah. Do you feel that is a future direction also in your area or your therapeutic area, that it will shift a little bit more towards a sort of a personalized medicine, that we check out everyone's genomic innate immune response? Is that a future possibility, or is that driving it too far?
There is. We don't know, of course, yet. But for example, genetically regulated immunotherapy is already happening in cancer, for example. We assess the mutations that take place in the cancer cell, and depending on that, we apply specific types of either
directly
immunotherapy
targeted.
or chemotherapy, which is directed for the special mutations of the patients. We can envisage a moment then this can be done also in infections. At this moment, the patient stratification that we try to do is based on the functional immunological variation in different individuals, because in the end, for the host defense against infection, several different mechanisms can lead to precisely the same change in the function of the immune cell. So we believe that actually stratification based on the function of immune cells themselves is likely to be more successful than just stratification based on one genetic or one epigenetic change. So we try to stratify the patients based on the immune function, and based on that to apply different types of immunotherapies.
Yeah. So are you also focusing research on the therapeutic pipeline?
Yes, we do.
Yeah.
Yes, we do also. So we are trying to do
Is
that
the outlook
in
hopeful?
Well, I'm always
We'll never know.
I'm also a very optimistic person, so I hope so.
Good. No one has the glass ball,
yes,
but
yes. But we are trying to direct our research in two directions, basically. One is trying to improve the efficacy of vaccines. We would try to integrate trained immunity in the classical vaccines, which are inducing T and B cell responses, which are very important. So just to be clear, the capacity of the vaccines to induce memory T and B cells is crucial, but we would like to build into the classical way of making vaccines also the capacity to train also the innate immune cells to make the vaccines even more effective. So this is one direction. The other direction is try to build this type of immunomodulation in severe infections, and especially we ourselves are working in sepsis, but I know that there are people who are thinking to do similar things in tuberculosis and in other diseases, in HIV, for example, and so on.
Yeah. Well, I can imagine this is going to be a very rich keynote lecture with enough information for people who want to know more about the immunity and infections. Thank you so much, Mihai, for coming here and taking the time on this big day for you to explain a little bit more about your keynote and your topic of interest. And thank you, viewers, for watching us. Check out the keynote lecture. It is this afternoon at 4:15 in Hall A. So that is definitely worth attending. We're taking a short break. We will be back at the clock of 12 for our hugely popular program element, the Lucky Wheel of Fortune. Your opportunity to win some really cool ESCMID prizes that you can either pick up here at the TV studio during your stay at the Congress here in Barcelona, or if you wish to attend online, that is also possible, and you can have a shot at winning some prizes which we will send to you after the Congress. So please make sure you follow the app and the platform in which the QR codes will be disseminated, and enter via the QR code, and I will hopefully see you then here live with me in the studio, and I will hopefully make a lucky spin for you at the Lucky Wheel of Fortune. So see you again at noon.